Okay. Assalamu alaikum. Today we want to talk about chapter 7, the behavior of proteins, enzymes, mechanisms, and control. And these are the sections that we are going to cover. From section 1 to 5, section 6 and 7 are omitted, and then we continue with section 8. Now, to talk about allosteric enzymes. Now, in the previous chapter, we talked about enzymes that obey the Michaelis-Menten kinetics, where they produce a hyperbolic type of curve when we plot velocity versus substrate concentration. But today we want to talk about other enzymes, allosteric enzymes that behave differently, enzymes that give us the sigmoidal type of curve when we plot velocity versus substrate concentration. And usually these enzymes or proteins are oligomers, which means they consist of more than one subunit, and they tend to change the enzyme's activity by changing the conformation of its quaternary structure. Now we want to introduce something called allosteric effector, which is a substance that changes the behavior of an allosteric enzyme by binding to it. This allosteric effector may be a substrate, a positive allosteric effector, or a negative allosteric effector. The enzyme we want to talk about is called aspartate transcarbamylase. This ATKs and hemoglobin, they exhibit cooperative effect caused by sudden changes in quaternary structure. Allosteric enzymes have a different response to the presence of inhibitors from that of non-allosteric enzymes. And to talk about the enzyme called ATKs, this enzyme catalyzes the condensation between carbamyl phosphate and aspartate to give us a carbamyl aspartate, and then there are a series of steps to give us the final product called CTP. Now, this final product called CTP, when its concentration reaches certain level, it inhibits the enzyme ATKs, and the mechanism is called feedback inhibition. As you can see here, this is called feedback inhibition. So CTP is an allosteric inhibitor of the enzyme ATKs. And to illustrate this, this is a schematic representation of how feedback inhibition works. Now, if you have this from enzyme 1 to enzyme 7, and then we have the final product number 7, goes all the way when its concentration reaches a certain level, go all the way to inhibit enzyme number 1. Okay. Now, let's talk about cooperative behavior of allosteric enzymes. As we saw, ATKs catalyze the condensation of aspartate and carbamyl phosphate to form carbamyl aspartate. And we said sigmoidal curve is produced for allosteric enzyme. Aspartate is here the substrate for which its concentration is varied, and the concentration of the second substrate called carbamyl phosphate is kept constant. How? At high levels, which means it is saturated. Now when we plot, this is the plot between velocity and substrate concentration, and you can see here the sigmoidal type of curve. Now when we add the inhibitor, as you can see, this is the control, no ATP or CTP, the blue line. Now here when we add the inhibitor, the curve shifts to a higher substrate concentration and becomes more sigmoidal at this high concentration of substrate. Whereas when we add an activator called ATP, the curve shifts to a higher velocity at lower substrate concentration. So we can say that ATP is an allosteric activator and CTP is an allosteric inhibitor of the enzyme. Now let's look at the organization of ATKs. It consists of two types of subunits, catalytic subunits and regulatory subunits. بعض الكتاليتيك we have two trimers as you can see, one above the other, and the regulatory are 3 dimers. Now, when the catalytic subunits, however, can be separated by adding an organic compound called para-hydroxymercury benzoate. Now, even if we do this separation, the enzyme still catalyzes the reaction, but it loses allosteric control. There are 2 types of enzyme inhibition mechanism, the K systems and the V systems. Now, any enzyme that belongs to the K system, when we add an allosteric activator or inhibitor, what changes here is the concentration, substrate concentration that yields 1/2 Vmax, and we call this K 1/2. You see, this is the same as Km in the Michaelis-Menten approach, K half. So for K system enzymes, K 1/2 is changed when we add the allosteric inhibitor or activator. And our enzyme ATKase is an example of a K system. However, if an enzyme belongs to a V system, what changes is the Vmax, the maximal velocity upon the addition of inhibitors and activators. Also here, again, the key to allosteric behavior is the existence of multiple forms for the quaternary structure of allosteric enzymes or proteins. Now, as I said before, allosteric effector refers to substrate, inhibitor, or activator that binds to an allosteric enzyme and affects its activity. There are 2 types of allosteric effects. We have homotropic effects and heterotropic effects. Now, in homotropic allosteric effects, these are the effects that occur when several identical molecules are bound to a protein. Remember, with hemoglobin, which binds 4 oxygen molecules, binding the first one facilitate binding the remaining of oxygen molecules, and all of them are similar, then this is homotropic allosteric effect. Also, the same applies for ATKase when the substrate aspartate binds to it. So these 2 are examples on homotropic allosteric effects. Whereas in heterotropic allosteric effects, these effects occur when different substances are bound to a protein. Remember, in ATKase, the substrate is aspartate, an amino acid. The inhibitor CTP, which is a nucleotide, different, and the activator ATP is also a nucleotide. Both of these are similar to each other, but they are different than the substrate called aspartate. So this is called heterotropic allosteric effects. Now, there are 2 models to explain the behavior of allosteric enzymes. One is called the concerted model, or it is also known by its discoverer, Monod, Wyman, Changix in 1965. Now, description of allosteric activity in which the conformation of all subunits change simultaneously. And this is the meaning of concerted. When a change occurs at one subunit, then a similar change must occur in the other subunits at the same time. Here we are concerned with the concerted model only. Now, in this model, the protein assumes one of 2 conformations. Let's see here, the R conformation and the T conformation. These 2, they exist in equilibrium with each other, and we define here something called L. L equal T divided by R, and L is assumed to be large. And this is the case because in the absence of the substrate, most of enzyme molecules are in the T state, in the T. طبعا R for relax, this binds substrate tightly, and T for tight or taut, this binds substrate less tightly. Now, when the substrate is added, it binds to the R form, of course, and the equilibrium shifts to the left. Now, according to Le Chatelier principle, here, when a system at equilibrium is subject to stress, then it goes in the direction to relieve that stress. So more T is converted to R, and notice that the value of L will decrease, of course. Also here we want to define two things: the dissociation, the enzyme substrate complex dissociation constant Kr, and the enzyme and the enzyme inhibitor dissociation constant called Kt. Now always Kr is less than Kt. What does this mean? It means that the substrate binds better to the relaxed form, and the ratio Kr divided by Kt is called C, and when C equals 0, it means that Kt is infinite, and this means that the substrate will not bind the T form at all. This is the explanation. Now again, in the concerted model, how the concerted model is used to explain the sigmoidal type of curve. Here, notice here in this figure, what we have the Monod-Wyman-Changix model for concerted. Here we plot Y, saturation versus substrate concentration. At constant C, we notice that as L, the ratio of T to R increases, as L increases, the curve becomes more sigmoidal. Notice here, the red one is hyperbolic, and as L increases, the curve becomes more sigmoidal. Now also, the second part refers to plot of Y versus substrate concentration at constant L equal 1000. Now we notice here that the level of cooperativity is based on the affinity of substrates for T or R form. When Kt is infinite, cooperativity is high, as shown in the blue line here. Cooperativity is high. As C, the value of C increases from 0 to 0.04 to 0.1, the curve becomes less sigmoidal and more hyperbolic. Okay, so these are the 2 points you should know about this plot. The first one, as L increases, the curve becomes more sigmoidal at constant C equals 0, and at constant L equals 0, as the value of C decreases, the curve also becomes more sigmoidal. Now in this figure, this figure explains the effects of binding activators and inhibitors with the concerted model. An activator usually a molecule that stabilizes the R form, and an inhibitor usually binds to the T form and stabilizes it. Now here, as in the model, we have a dimeric protein. This is a dimeric protein that can exist in either R or T form, and the protein can bind 3 types of ligands. The first one called substrate, the second called activator, and the third called inhibitor. Now, when the activator binds to the R form, the equilibrium shifts to the left. When the activator binds to the R form, the equilibrium shifts to the left, and more substrate molecules are required. No, less substrate molecules are required to reverse the equilibrium, and cooperativity will decrease. So what are the effects of adding the activator? Decrease in cooperativity, and also the value of L decreases too. whereas when we add the inhibitor binds and stabilizes the T form. Now the equilibrium shifts to the right, and now more substrate molecules are required to reverse the reaction. So cooperativity increases. Also, the value of L increases too. Now, if we look here at this plot Y versus substrate concentration, notice in the presence of no activator or inhibitor, sigmoidal type of curve. When we added the inhibitor, as I said before, the curve becomes more sigmoidal and shifted to higher substrate concentration, whereas when we added the activator, the curve becomes less sigmoidal, more hyperbolic. And here you can see the value of K1/2 here. لاحظوا the highest value is in the presence of the inhibitor, 10, and the least value in the presence of activator. So K0 means this value, which is equal substrate concentration at 1/2 Vmax, increases in the presence of the inhibitor and decreases in the presence of the activator. Okay, now we want to talk about control of enzyme activity by phosphorylation. Many enzymes have at the side chain hydroxyl groups, serine, threonine, and tyrosine, and this hydroxyl group can be, however, phosphorylated. By what? By ATP. And upon this phosphorylation, which is also called covalent modification, can convert an inactive enzyme into an active one, but not always, because for other enzymes, dephosphorylation activates the enzyme. Now, examples. This is how it looks like. You have here a serine residue become phosphorylated, threonine residue become phosphorylated, and tyrosine residue become phosphorylated. Now, an example on that is the so-called phosphorylation of the sodium-potassium ion pump, where the pump itself, as you can see here, ATP donates a phosphate group to an aspartate residue 369 as part of the mechanism, and this causes a conformational change in the enzyme. And this mechanism, 3 sodium ions enter or goes outside and 2 potassium goes inside. The details are not required at this stage, only that you should know that the sodium potassium ion pump is an example of an enzyme or protein that can be controlled by phosphorylation. Proteins that catalyze these phosphorylation reactions are called protein kinases, a class of enzymes that modify a protein by attaching phosphate group to it. And the usual donor is ATP. So any enzyme where its name ends with a kinase, it means it requires ATP. And ATP is the source of the phosphate group. These enzymes, they play an important role in metabolism, as we will see. A second example is the glycogen phosphorylase. Glycogen phosphorylase catalyzes the initial step in the breakdown of glycogen that is stored in the muscle. Exists in 2 forms: the phosphorylated form called glycogen phosphorylase A, and the dephosphorylated glycogen phosphorylase B. Usually the A form is more active than the B form. However, the 2 forms of the enzyme respond to different allosteric factors depending on the tissue type. Glycogen phosphorylase is thus subject to 2 types of control. allosteric regulation and covalent modification. Of course, the covalent control here is the phosphorylation, and the non-covalent or the allosteric control is where these allosteric factors tend to either activate or inactivate these different forms. So the net result that the A form is more abundant and active when phosphorylase is needed to break down glycogen to provide energy. But generally speaking, we cannot predict whether phosphorylation will increase or decrease the activity of an enzyme. Now, zymogens. A zymogen is an inactive protein that can be activated by specific hydrolysis of peptide bonds. The proteolytic enzymes trypsin and chymotrypsin, they provide a classic example of zymogens and their activation. Their inactive precursors are trypsinogens and chymotrypsinogen are formed in the pancreas, where they would do damage if they were in an active form. However, in the small intestine, where their digestive properties are needed, they are activated by cleavage of certain peptide bond. So again, we want to talk about chymotrypsinogen as a zymogen of chymotrypsin synthesized and stored synthesized in the pancreas, in the liver, stored in the pancreas and released into the small intestine. It consists of a single polypeptide chain of 245 amino acid residue cross-linked by 5 types of disulfide bonds. However, when secreted into the small intestine, the digestive enzyme called trypsin cleaves a polypeptide bond from the N-terminal end to give the chymotrypsin. Now, if we look at this figure, which shows us the proteolytic activation of chymotrypsinogen, we notice that here we have, as I said, consists of 245 amino acid residue. This is the inactive zymogen with 5 types of, you see these yellow, these are the disulfide bonds, 5 of them. They occur between 2 cysteine residue each. Now, when this enzyme or chymotrypsin is secreted into the intestine, it is acted upon by the enzyme called trypsin. Trypsin is very specific enzyme. It cleaves at the C-terminal of 2 amino acids, arginine and lysine. So it cleaves here on the right of, this is called the C-terminal. This is the C-terminal of carbon 15, and here you see a break here. This is called pi-chymotrypsin, although it is an active enzyme, but it is not the final active form, which again undergo self-digestion at the following position: leucine 13 here, tyrosine 146, and asparagine 148 by pi-chymotrypsin to release these 2 dipeptides and generate the so-called alpha-chymotrypsin. This is the active form of the enzymes. The 2 dipeptides fragments cleaved. The final form of the enzyme has 3 polypeptide chains, as you see here, but they are, however, connected together by 5 original and still intact disulfide bonds. But now we notice 1, 2, 3. 3 of them are intra, means within the same chain, and 2 inter disulfide bonds. Inter means between 2 different chains. So whenever we read the word chymotrypsin, it means alpha chymotrypsin unless otherwise specified. Now, the protonated amino group of isoleucine residue exposed by the first cleavage. Here, here we have at this position we have isoleucine residue that at this where here we have an amino group with a proton. Now this protonated form is involved in ionic bond with the carboxylate side chain of aspartate residue 194. Now this ionic bond now is necessary for the active conformation of the enzyme because it is near the active site. Chymotrypsinogen here lacks this bond, therefore it does not have the active conformation and cannot bind the substrate. This is the end of lecture